Hi, everyone. I'm Janja. I apologize in advance for my voice today. It's just at that time of year, so hopefully you can all still hear me okay. So we talked a lot already about phages in general today. So during this presentation, I will present the various fields where phages can or are already being utilized. And I'll focus on agriculture, food safety, environmental decontamination, and of course, veterinary and human medicine. To start off, I want to talk about the very wide range of possible applications that phages have for use in environmental decontamination. This can be anything from wastewater treatment and soil remediation, where phages can be used to remove certain bacteria from these environments, to very specific cases, for example, hospital environment cleaning, because as Hans-Jörg already mentioned, hospitals are environments where we are most likely to find the so-called superbugs, so bacteria that are highly resistant to antibiotics. And if we could use phages not only then for treating, but already for reducing the amount of these bacteria in such environments, we could greatly limit the spread of AMR bacteria. Currently, there are no products yet on the market for use in this field, but there are several in development. The next possible application is the use of phages in agriculture, because here different pathogens cause over 40% yield loss of different crops every year. And this translates to an estimated $290 billion of annual economic impact. In addition to this, the EU's Green Deal also aims to reduce the amount of synthetic agrochemicals used by 50% by the year 2030, which means that right now there is a great demand for alternatives such as microbial or phage-based products to be on the market. Phages can be delivered in different ways, which we already briefly touched on in the Q&A. So they can be used for soil drenching, which basically means phages are added into the water that is used for watering the plants. They can be sprayed onto the plants. In certain cases, they can already be used for coating the seeds before germination. The advantages of phages in this field is that they are highly specific, meaning that they will only impact the specific bacteria that they are targeting. They will have no impact on the crops themselves or any surrounding microbes. They are also very safe to use, especially compared to traditional agrochemicals, and they are also completely safe then for consumer if they were to remain on the plants. They can be used for a very wide range of applications. One is the Pierce's disease of grapevine, then different diseases caused by pathogens on tomato, citrus, and peach, and also potatoes and different leafy greens. In the US, there are already quite a few products on the market. These mainly target tomato and or pepper bacterial spot and speck. And also products that target pathogens that cause citrus canker and apple or pear fire blight. Unfortunately, in the EU, there is currently no product yet on the market, but there are some in development. So the path from agriculture then moves to food safety, where we were quite surprised by the high numbers, but the World Health Organization estimates that there are around 600 million foodborne illnesses and almost 500,000 deaths annually caused simply by by consumption of unsafe foods, so food illnesses. And this also means an estimated $110 billion of annual economic impact. These foodborne illnesses can be caused by different pathogens, which are listed here. The last two, especially like Clostridium or Staphylococcus, can be especially dangerous if they produce a toxin, which can mean more severe reactions in the consumer. There are different delivery systems where we can incorporate phages. They can either be added directly into the food, or they can be used during food processing, which is maybe more closely also then linked to, for example, the field of environmental decontamination. And they can also be used by incorporating them into the food packaging, which is called active packaging. So they are slowly being released during the storage of the food. Again, also here, the advantage of the phages is that they are highly specific, and they are also able to infiltrate biofilms. Biofilms are a way that bacteria can, in a way, hide inside these matrices, but since phages have the ability to infiltrate them, they can still be effective against the bacteria, even if they form these biofilms. And one very important advantage of phages in this field is also that they have no impact on the characteristics of the food, whereas other processing such as pasteurization and similar processes always in some way affect the characteristic or the quality of the food. So as I mentioned, there are different possible applications. Some can be even more closely linked than to either agriculture or, for example, because we can already treat animals on the farm level, the so-called pre-harvest application of phages. This is mostly to control the Salmonella and E. coli levels. Then we can have post-harvest decontamination of the food themselves. This is mostly used to treat ready-to-eat meat, leafy greens, and dairy. And then also it can be used for food contact surface sanitation and food processing aids, said that the phages can be added directly onto the food. In this field, there are quite a few topics, quite a few products already on the market. In the US and in Canada, we have FDA and USDA approved products against Listeria and Salmonella, and also product against E. coli, which is used to treat red meat and produce. In the EU, there is only one product available, and that is to treat Salmonella in poultry. Interestingly, there are quite a few products also available in India, China, and also one in South America. They are used for very similar products than in EU and in the US, with the exception that in India, they also have a product that is used in shrimp farms against Vibrio. For our last two topics, we will discuss phage applications in veterinary and human medicine. In veterinary medicine, the majority of work and products are being developed for animals that have an economic impact. So this is for poultry, cattle, pigs that are used for human consumption. There are, of course, also some cases where phages are being used to treat companion animals, so dogs and cats. But for now, those are mostly single isolated cases. As I said, most of the applications are being used on these animals, where I think that the One Health approach that Hans-Jörg already mentioned can really be well demonstrated, because if we can decrease the pathogens with phages on this level, not only we can make food more safer for the consumers from the standpoint of foodborne illness, we can also reduce the amount of antibiotics that we need here, which means less prevalence of antibiotic resistance and bacteria. Regarding products on the market in this field, there is one product that is currently approved for use in EU, and it is a product that's used in aquaculture. So our last topic is, of course, phage applications in the so-called phage therapy, so for use in human medicine. We already discussed today that the status is already quite critical in this regard, and the projections are looking even more grim. So not only the large amount of deaths that are predicted to be caused by AMR, but also an estimated $1 trillion in extra healthcare costs will be needed by 2050 just to cause AMR infections. The most common indications where phages can be used are chronic infections, so wound infections and prosthetic joint infections. Then also respiratory infections, especially infections that are connected to, to cystic fibrosis or ventilator-associated pneumonia. Then urinary tract, gastrointestinal, and ear, nose, and throat infections. But not only this, phages are also already being used for treating acne and similar issues. So they're being used not only specifically in medicine, but there are some application also for cosmetics. There are currently two main approaches to phage therapy. One is a more personalized approach, and the other is the conventional approach. So the personalized approach encompasses compassionate use cases and magistral products, and this is mainly being used for now in the EU. Compassionate use is where for critically ill patients, no approved treatment is working anymore, and in those cases, non-approved drugs can be used. There have been hundreds of such cases where phages have been used as a last resort with really amazing outcomes, because in over 85% of the cases, there was pathogen eradication with these really critically ill patients. So the other approach to this personalized medicine is magistral products, and this means, as any other magistral product, that it is prescribed per patient specifically, so that it's prepared in the pharmacy for a specific patient. And then, of course, the conventional path is where phage formulations are pre-developed and pre-approved through clinical studies. Below, you can just see a quick summary of which pathogens different studies are targeting. And of course, here we are very happy to see that some of these studies are already progressing to phase 3 studies. So it shows good progress for the future. So if I could just quickly summarize all of these applications, we're very happy to see a lot of products in different fields already on the product, are already on the market, and many others in development. What we do see is that EU might be slightly lagging behind other countries. So in the future, we would like to see also more products developed and approved also for use in the EU. That's all.
Perfect. Thank you, Janja, very much for your presentation. yeah, as you will see, a lot of applauses, thumbs up, really exciting and very promising, especially I think the last slides you showed also on human medicine. It's really indeed promising. The first question that we received for you was whether you could maybe use some phages to clear up your throat. What could be your response?
Yeah, I think I'm colonized by a different virus, so I don't think phages would help.
Okay, but to conclude, you have raised indeed very important issue. It's actually why, comparing to other parts on the world, Europe is still lagging behind with regards to access to phage-based products and therapies. But actually we have also a specific question about this in the chat that Hans-Jörg has already replied, but I think we'll be also happy to hear your perspective to see if it's similar. So the question, let me read it for you, is the following. Why is it that the EU still doesn't have or yet is developing phage products for crops when compared to the US, where they are doing 3 already? Would it be that the crop pathogens have different characteristics?
It's possible, but I think that it's maybe more regulatory hurdles that they are not approved yet in the EU.
Indeed. So I would say you're in line with Hans-Jörg, and we as PhageEU also, we are doing our best to work together with decision makers, also agencies, and big thanks to European Food Safety Authority, European Medicines Agency for their openness and interest in phages. We know it's not so easy to develop guidelines so that we can facilitate the authorization processes. There is also need for experts who know the phage technology. But we can, I think, also admit that we see an increasing interest in the phage technology among decision makers in the EU. So we hope that the situation will improve in the near future. Recently, we also have seen some funds being earmarked within the Horizon Europe 2025 work program, some funds devoted to phage therapy. So it's indeed very, very promising. There is another question. One participant is saying, I think one of the hindrance for the application of phage therapy is product specification, their management. What is the best solution to solve this issue? I understand it's maybe the need to adapt the cocktails. So once you get an authorization for a certain product, you also need to adapt it during the life cycle. At least this is how I understand this question.
Yeah, I'm not sure I understand. So the product specifications during the life.
We can ask maybe to maybe the person who asked the question to maybe try to be a bit more specific. Unless Hans-Jörg, you have any idea how to respond to this question? You have to unmute yourself. Next to unmute.
Okay, you hear me now?
Yes. Yes, now we can hear you.
Okay, which question would you like me to address? About the opportunistic pathogens that can come up?
No, the question that is not too clear for Janja, that one of the hindrance for the application of phage therapy is product specification / management. What is the best solution to solve this issue?
I'm not the best person to answer that.
Yeah, maybe I just regarding the life cycle, I wasn't sure, but just regarding the specification, then in the past, I think that may have been, but I think now the product specifications should be now known. So just the limits of what we need to reach for identity purity. So that should not be a hindrance now for phage therapy.
Okay. There is a question, maybe not very much linked to the applications, but in general about phages. So you can maybe see who of you would like to address it. So the question is the following. If we restrain the growth of specific bacteria using phages, is it possible to give other opportunistic bacteria the way to flare up and become more harmful?
Would you like me? Or
you
or
can try,
Janja?
you can try, yeah.
If you make an intervention in a microbiome, that's always the risk. So you suppress one bacterium, another bacterium might grow up into this niche. But why it should become more pathogenic is not logical to me.
Yeah.
So there's no link to that. Of course, if you have 2 or 3 highly pathogenic bacteria somewhere and you only eliminate one, then you have 2 pathogenic bacteria in, in your system still. And so you have to think about how to eliminate those. The fact is that with bacteriophages, you just specifically eliminate one you target with the bacteriophages.
Okay, and maybe last question, which is quite long, and we still have 9 minutes left. We have a question actually from our scientific member. Thank you for the excellent presentations. I would like to ask about the regulatory perspective in the European Union. What are the main differences in the evaluation focus between phage applications for farm animals, for example, as feed additives or veterinary use, and food-related applications such as surface decontamination or food processing? Specifically, which aspects are emphasized differently in the EU review process, for example, genomic, safety, environmental risk, or human exposure?
I think that is maybe more Hans-Jörg's expertise, but just in general, I would
could
say
not
that
really follow. I would need to read it.
Yes. We
But
also
just-
have, yes,
yeah.
please, Janja, please.
just in general, for there are different aspects that would need to be how do I
Taken
say
into
that are
account.
important in different fields. For example, for the use in farms, allergens are not that important, whereas for use in food, you have to be really careful how you control allergens, even in phage production to be used. So each field has its own specifics, but I don't think that either one of us is that confident to list exactly what the differences are.
Okay, no problem. We also have with us some other PhageEU members. Maybe anybody would like to take the floor and reply to this question? If yes, please just unmute yourself and intervene. I see Frank wants to say a few words.
Hey, yeah, hi. So Frank Smaker from JAFRAL, and also yeah, I would just add to what also Janja already started. So they're not since they're not specific guidelines, or for veterinary now it is, and for human it's coming. There are some specification, but as Jan already said, it really needs to be looked from the application point of view. It's funny, right? Because in case of the human therapeutics, which you say is the highest grade, but you don't care so much of the food allergens, where in if it's for food that you do. If as it was questioned here, so more from the phage perspective, what kind of phages you have to have or select. There is in all cases, it is important that when you're using the phage for whatever application, the fact is that you're putting that in the environment. So because you're putting in that environment, you have to be aware that that if that multiplies, that so in either way, it's not good to have, for example, isogenic, if that's possible, whatever the application. And that's why, at least in Europe, it's also the consideration of GMO, stuff like that. So this is, at least from the, let's say, first phage characteristic point of view, but then as said, for the final product as a quality perspective, yeah, can be different considerations. I hope that helps.
Perfect. Thank you, Frank. And I also see that Luis Rocha from LX Bio also intervened in the chat, trying indeed to respond to some questions. Indeed, more and more questions are coming, but unfortunately we will have to, I think, close our session. But don't worry, as I said, we will have a careful look at these questions and we will try to address them in the future. For sure, I think it seems that the regulatory issues, regulatory hurdles, and the regulatory pathways, that's something that is of interest to many people. And we will then probably try to address it also during one of the next sessions. But in the interest of time, I think I would like to first of all thank our experts for their great presentations. Thank you very much for your time preparing your presentations and for being with us today. I would also like to thank our participants for their time and for their active participation. We had really a lot of interesting questions, around 80 people with us during the whole session. So it's a great result, and it confirms the great interest in the phage technology. So as I said, this session will be published on our website. So you will be able to watch it and to follow the presentations. You will also receive after this meeting a satisfaction questionnaire where you will be asked to indicate topics that are of interest to you. So really, please take your time and please specify the issues you would like to see covered in the future sessions, because we would like to organize the next sessions of PhageEU Academy according to your needs and your wishes. I would also like to take this opportunity and invite you already for 20th of November, because we will be organizing another online session at the occasion of AMR Awareness Week. On the 20th of November in the afternoon, you will hear from some of our PhageEU members. They will be sharing some recent developments and innovations in the phage technology. So once again, thank you very much. I wish you a happy World Phage Day, a very good end of the week, and I look forward to meeting you soon again. Thank you very much. The session is closed.